Thank you very much, Peter, and thank you to all organizer for organizing this. That's great. And for me, it's great to be back in Tübingen. It was one of the most important and most fun times of my life. Mine and my wife, Barbara, over the 10 years in Tübingen in the 70s. I'll speak about a little bit of scientific history, and of course, what I will say is closely intertwined with what Kuno already said. So you may hear some of the same things. Werner after the war survived in Tübingen repairing radio, eventually making it to a PhD, and some of his advisors were Lauer and Ruska, who developed the electron microscopy and got a Nobel Prize for it. And so the story that you already heard went after that with the creation of a research group in Tübingen, and then director of the Abteilung, and started a journal for Biological Cybernetics, and then created this institute, 68. And I think I arrived here in 71. These were the 4 directors. Maybe a little bit younger, but that happens to everybody, I guess. What about? I think it was interesting that while he was in Germany, he was a German professor, and in the best of the German tradition at the time. When he was in the USA, was a hippie, absolutely different person. I remember some trips in the desert when we borrowed camping equipment from the widow of Max Delbruck, and had a lot of fun, got a little bit drunk in that valley, were rescued because we got stuck, and all very lot of fun. He was fantastic with young people like me. He introduced me when I was, 25 or so, to essentially all the most important people in neuroscience at the time in the US. This is a picture of the one, which was a kind of very special club, the Neuroscience Research Program. And if you look there, there is, look at the bottom here, but also Cowan, anyway, it was Werner was part of this club, and he introduced me to it early on. It was a great gift to a young scientist. As Kuno mentioned, the main argument for starting in the Max Planck Institute for Biological Cybernetics was to study nervous systems, a different level of organization, from behavior to what we would say the algorithms, to the neurons and synapses and their circuits. So these three levels. And this three level story is something that has had, I'm told, pretty big impact in different areas of neuroscience, especially cognitive science. Was popularized by David Marr in his book Vision. The argument is that if you want to understand the complex system, it could be a computer or a brain. There are different levels. In the case of a computer, they are almost independent of each other. You can understand the computer, for instance, in the sense that you are an expert of PowerPoint. That's your understanding. It's powerful. You can do things. You can understand it at the level of how logical gate in the computer works, or how transistors are assembled in order to make logical gates. And you can be an expert on this and know nothing about how to operate PowerPoint. So these are independent level of understanding, each one powerful in its own. And so that's an obvious way to distinguish what you want to do or to discover or to study. Something to keep in mind. It's somewhat arbitrary, but it's interesting that this argument that was popularized by David Marr in his book really came from a paper that I co-wrote with David in 77, and which was by itself very similar to the argument that we had in a paper on a fly with Werner Reichardt, and that of course came from the original argument for the Max Planck Institute of studying systems like the fly at different levels of organization. Now, I want to go through this. In my experience, the study of the fly is essentially cognitive neuroscience in the fly. In 10 years I was here with Werner. So it started, I arrived at 71. This is, by the way, David Marr. Werner Reichardt is on the extreme right. And here is Buchner. Yeah. After playing some wonderful music I think in the guest house at the time. It's Werner with Jonathan. It's Johannes. Sorry? Yeah. So there are essentially the three levels where studying behavior, and particular behavior in the fly, or fly, Musca domestica as well as Drosophila do fixate stationary or moving objects while flying and track other objects, but for instance, they chase other flies. And this is really a kind of phenomenological study of this, little bit like thermodynamics in physics. You don't know which neural mechanisms are used, but you can still describe in quantitative and predictive terms what's going on in the behavior. And then at a lower level, you can try to understand how motion that, for instance, is important for fixation and tracking, how is that computed from the visual inputs to the eyes. This is essentially algorithms, and I'll describe briefly a part of it, the correlation model that Kuhn already referred to. And then the bottom level is the one, of course, of what are the circuits that implement these algorithms. Difficult to read, but this was 71 June was the offer I got from Werner Reichardt to come here originally for 2 years or 1 year, and then I stayed for 10 or so. So level of understanding. So this was the vision book. This is an image of, there is David Marr, on the back is Francis Crick, you cannot quite see him. And the vision book, a computation investigation to the human representation processing of visual information, which was published after David died, is the one where this approach, this idea of different levels was described and popularized. And as I said, this argument came from this previous paper indirectly from the Max Planck Institute here. So this is the beautiful eyes of Musca domestica. Kuno is the one who did beautiful work on the optics of the fly, and this behavior of the fly can track stationary objects, can chase other flies, was studied for several years by Werner. This was already going on when I arrived here in 71. He had built essentially virtual reality simulator at the time. Remember, there were no computers, no internet, essentially no screens. So the virtual reality was a real cylinder on which a pattern was put, like a black vertical stripe. Thank you. Thanks. And the fly was suspended from this device that was measuring torque, designed by Carl Goetz. And so a tiny torque. The fly, if you put some paper on the leg, grabs the paper, stop flying. If you take it away, start flying. So the body of the fly was glued to the compensator, this device, which was keeping the fly perfectly fixed, but measuring the torque. And later on, you could measure other things like lift and other dynamic effects of the fly. So there was a simulation of the flight dynamics, like friction and inertia. And so it is as if the fly was believing she was freely flying. But what happened in reality is that the torque the fly generated was transformed in motions around the vertical axis of this cylinder, so as if the fly were able to rotate. So it's a virtual reality environment, but in which you could study quite easily what's going on. And if, for instance, there was some black pattern, like a vertical stripe against a bright background, the fly was trying to fly towards it. And even if that object was, for instance, moving. So this was the fixation behavior, and through it, so you could measure this quantity as if in a free flight situation. Suppose this is the direction of flight. There is an object here, and you can measure the angle of the between the direction of flight and the object, and this was essentially the position on the eye of the fly of the image of the object. So this was the error angle, and the fly flying towards the object was trying to minimize this error angle, put psi to 0. So through a lot of experiments, Werner had come up with an equation we refined together, in which there is a part which is just the dynamics of the flight, which was simulated in this virtual reality environment, and then there were other terms that came from the fly. There was a kind of search term, it was like a noise term, roughly Gaussian in the torque. And then there was another term which was kind of a correction to the error. So the fly was trying to generate a torque that was going to set psi to 0, this error angle. And there was another term that depended on the relative motion of the object relative to the motion of the fly. This term is what the target may be doing, which was controlled by the experimentalist. And so this is a stochastic differential equation because of this term, this search term generated by the fly, which is noise. We know only variance and so on, and some stochastic properties. And but it can be solved under certain assumption, and so we had equations that could really describe the behavior of the fly in the simulator under different conditions. One question that was open is how well would that this kind of model and describe what a fly is doing in real life, in real behavior, not in the simulator. And in the 80s, Christian Wehran started doing the experiment in free flying flies, flying in a cage, and analyzing frame by frame, reconstructing the 3D trajectory of the flights. And so this was kind of the setup in which we wrote. At the time, even finding a high-speed camera was not so easy, but like 100 frames per second, and then this could retrieve the XY position of flies, and to a mirror here also the Z position, so XYZ of for frame by frame, to reconstruct the 3D trajectory of flies in this environment. And flies were chasing each other. It seems mostly to be sexual behavior. Mostly it's males chasing females. Here's an example. There is a chase, probably lasting 1 second or 2 seconds or so. This is a stereo pair. You could fuse them if anybody can. You can see these trajectories in 3D, so they're kind of interesting dancing in 3D. And this is a simulation of the chasing fly. The fly is chasing I think one of the two is the chasing flight. So I think the first flight is the asterisk, and the other one is the chasing flight. And anyway, so you put in this equation, in this term, the trajectory of the chasing of the original flight, the flight was chased, and this equation produces the trajectory of the chasing flight. So it's difficult to gauge here, but there is a pretty good agreement between the experiment and the prediction of the model here. In 3D. So, and there were other experiments. For instance, these were done together with Gadi Geiger, in which you have something that in the behavior of the fly that is a bit similar to our so-called Müller layering. Yet we have perhaps you have the illusion that this is maybe longer than this, whereas in fact they have the same length. If you show this to a fly in that virtual environment I described, you see that for this one, the fly will mostly fixate in the middle around here. But in this case, it will fixate mostly around here and around here, jumping between the two. So another prediction from this simple prediction is that the fly could see, so to speak, illusory contours. So here you don't have a real bar, a real boundary, but you have an illusionary one, same here. And if you look at, show the fly, these two patterns, you see the fly will mostly fixate around here and around here. So, so we had a simple model theory that seemed to be quantitatively okay in predicting behavior of the fly. There are many caveats. We did not take into account the fact that, for instance, the head of the fly moves relative to the body in real life, did not. And so it's not the final word, but not a bad approximation either. So, but then this was the level essentially of the behavior, describing the behavior, kind of thermodynamics of the fly. Then you can ask, okay, you know, in those equations, I need to have, for example, psi, the position of the target on the eye. Now, this is not given to the fly. It has to be computed from the images. How is this done? Motion, there was a psi dot, the derivative in time is the velocity, essentially, of the target on the eye of the fly. How is that computed? And we had already for motion, from work on the beetle that Kuno described, an algorithm for computing motion, the correlation model. That's essentially the algorithm. And so this was work that Werner Reichardt found out, whether Barrio came up with Reichardt detector correlation algorithm, and the same model seems to describe pretty well what happens not only in the beetle but also in the fly. And by the way, of algorithm that describes the psychophysics of motion perception in humans, mostly by Ted Edelson and co-workers. And this is essentially equivalent to the correlation model, formulated in a different way, but it's mathematically equivalent. So it's not only flies, but also primates, humans. And one of the first technique to stabilize the images of video cameras was really based on correlation-like techniques. It had an implementation in the connection machine at the time, 89, of this kind of algorithm. So this is one version of the algorithm in which you have high-pass filter, multiplication, low-pass filter here. That's very similar to a kind of logical version of it in which you detect motion by delaying this signal and then having this signal vetoing the signal coming here. This will happen if you move from the right to the left. The signal that comes here is delayed and therefore may arrive here depending on the velocity and the spacing of the photoreceptor at the same time as this signal. And in that case, this will be able to veto the signal from one. And so this way you can distinguish direction of motion, which is a nonlinear has to be a nonlinear operation. Another one, which is like we call the relative motion, and it's important if you got a fly chasing another fly, there is not only the image of the other fly, but also all the environment, which is moving because the first fly is moving. So you have to distinguish motion against a background, which is also moving. And, oops, let me see if I can do it. Can you see? No. Relative motion can destroy camouflage. And fly can do that, can see this. Question is how, and they can do that, by the way, not only in this case in which the background is stationary, but it's enough that the two move relative to each other. And so we also study essentially the algorithm underlying this, how can this be done, and came up with a model that looks very complicated, but basically consists of inhibition, lateral inhibition between motion detectors. You can think of a center surround machinery between motion detectors. Right? And as far as I know, this is still open for grabs in terms of the neurophysiology of it. Something for Axel to do next, your retirement. Okay. But we had a description of what's going on and measured various experimentally various predictions that seems all to fit pretty well. So we have examples of the phenomenological level, the level of algorithm for motion. I did not speak about position, that's fairly easy, but relative motion. But the next question was how do you implement this algorithm? Which neurons, which synapses, which biophysical mechanisms? You don't have multiplications really in neurons and synapses. You may have approximation of them, but which ones are actually used? So one proposal, and I'm sure that that we had at the time, but Axel will speak about what actually happens. But one proposal that we had with Vincent Torre was that this non-linear interaction, something similar to a multiplication or to a AND NOT gate or AND gate was actually implemented by the nonlinear interaction of conductance changes in a neuron. So in this, what happens in a neuron is that synapses don't really inject current, in which case you'd have linear behavior, but they change conductance or resistance to certain ions, with each one of which has an equilibrium battery, an equilibrium voltage. And so you may have, for instance, two synapses interacting with each other in a membrane, in a dendrite, and they may be both really excitatory, like just G1 and G2 by themselves change the depolarization of the cell, and in fact G1 has correspond to a battery which has a much higher voltage, and G2 to one which is an equilibrium close to the equilibrium potential of the cell. So even a large change in G2 which depolarize only a little bit, and in fact it may, in a real life, may hyperpolarize, but this is an extreme example. When they come together, essentially what G2 is doing is shunting G1, is opening a hole in the membrane, and so you have a nonlinear interaction. G1, the effect of G1, instead of being this big, because of the simultaneous presence of G2, will be much smaller. So this turns out that this is similar to multiplication. If you expand in a Taylor series and so on, it's essentially similar to multiplication. In this case, it would be this one, G1 times G2. So kind of reflecting this, and it's similar to a NAND gate. So this is a biophysical mechanism. This would be an example of biophysics of computation. We use biophysics to do computations. And as I mentioned, of course, we know that some of the big neurons involved here, I'm sure Axel will speak about it, right? And some of this are involved probably in certainly in motion, but also maybe relative motion. And in the case of relative motion, we still don't know which kind of circuits are at work. But in the case of motion, you will hear from Axel. This was really a proposal how this is actually happening in reality. It's a bit more complicated and even more beautiful. Yeah. And so, I'm finished here with this recollection of the work with Werner. I find always difficult to speak about Werner because for me it was one of the most important people in my life. He was a close friend, was my main mentor. I think I grew up as a scientist with him, under him, and he was a wonderful person, somebody you could always trust, somebody you could always believe, convinced of his beliefs. I have only great great memories of him and his life and his wife. So not much I can say more, but I can try to answer a lot of questions. The kind of things I'm doing now, like this is the book that I co-wrote. It's unfortunately it's only in Italian now, but will be translated in English soon by MIT Press. It's actually Brains, Minds, and Machines. It's about natural and artificial intelligence. And I'm not sure how this will connect, how it does connect to the brain, to the work that I did in Tübingen. I think we'll see. There is it's a question of whether the AI of today, large language models, and the approach they are using, which is essentially exploiting what mankind has produced over centuries in terms to create an artificial intelligence, whether this will continue to be successful as it has been or not. We have now systems like GPT-4 or Gemini or Claude 3 or so that are, I think, as intelligent as we are in terms of the old Turing test. I think they will all pass it. Of course, I also believe there is an infinite number of different forms of intelligence, so it's not surprising they are different from us. They are better in some respects. For instance, I can use GPT-4 to translate among more than 100 languages. I have one postdoc who speaks fluently 14 languages, but I think it's not usual. It's certainly not 100. But on the other hand, they're still rather stupid in other respects. Not clear they can really do abstraction, produce a theory, do induction, but maybe tomorrow. Now that's a bet, and I don't know, I'm 50-50 whether that will be possible or not. If it will not be possible, then I think one of the few alternative approaches to intelligence, artificial intelligence, is to go back to neuroscience. And after all, our own intelligence is the best example we have, and we can study it at different levels. And maybe from that, create real intelligence, artificial intelligence. So we will see. I think in any case, we need to understand human intelligence at different levels. And there are many reasons for that. One is because it is there are many great problems in science: the origin of the universe, the origin of life, the structure of space and time. But I would argue that the greatest of all the great problems of science is the problem of human intelligence. How it has, how evolution discovered it, and what it is. And so we need to solve it like we need to climb the Everest, or that's scientific curiosity. But there are also practical implications. For instance, if at some point we want to connect ourselves to the internet, to other machines, to increase our memory, so on, we need to know where to put the plugs. What is the protocols for communicating between machines with the brain? And if you, like some of the billionaires of today, really care about immortality or going to Mars, that's probably the best way. Download it yourself in a computer and you're fine. I'm just say I don't particularly. That's not the reason I want to understand human intelligence, but it's a practical reason for some people. But let me stop here and ask for questions if you have any. Thank you.